have been doing. So the first team you'll hear from today is SF Bay Area DIY Bot. Take it away, guys.
So we're the Real Vegan Cheese Project, and we're centered in California, San Francisco Bay Area, and we're centered around 2 community labs, BioCurious and Counterculture Labs. We have a very diverse team, ranging from PhDs and college grads to high school students and middle school students. And we're a purely crowd-based funded project, so here's our Indiegogo campaign to introduce our topic.
To make cheese, you need milk, and to make milk, you need mammals. But as we all know, farm animals aren't always treated as well as we'd like. Then there's the environmental impact of having 260 million dairy cows worldwide. And let's not forget that 60% of adults are lactose intolerant and can't eat many fresh cheeses. What if there was another way to make cheese without the problems? We're a team of citizen scientists and biohackers based in the San Francisco Bay Area, and we're working hard to reinvent how cheese is made. The big problems with cheese all have to do with where we get the milk, so we're going to replace cows with these little fellas. This is Saccharomyces cerevisiae, better known as baker's yeast. We're going to give baker's yeast the ability to make milk. Okay, maybe not quite milk, but at least milk protein, which is the important part of milk when it comes to cheese. Here's how it works. We're taking genetic sequence for milk protein from cow DNA and creating cheese protein genes that function in yeast. We then insert the cheese protein genes into living yeast cells. The genetically modified yeast will produce cheese protein as it grows, and raw cheese protein can then be extracted and separated from the yeast. To make real vegan cheese, we simply take the cheese protein and combine it with the 3 other main ingredients of cheese: fat, or in this case vegetable butter, lactose, or for our cheese a vegan sugar, and good old-fashioned H2O. Mixing these up makes a kind of vegan milk, and from this milk we'll use the same cheese-making process that has been practiced for thousands of years to make Gouda, Emmental, and even Parmesan. Our team includes experienced biotech engineers, computer scientists, and dedicated vegans. As with all scientific research, we can't be completely certain that this will work until we try it. But with your help, we're confident that we will be able to make vegan cheese protein, and with a lot of hard work, turn it into the world's first real vegan cheese.
Okay, so here's our process in brief. So first, we're going to search for the genes of interest in our genome. So theoretically, we can make cheese from any sequenced mammal. We're going to synthesize the DNA. We're going to insert the casein, or the genes of interest, into yeast using strawberry DNA as a carrier. We're going to express and purify casein proteins, which are secreted by yeast into the supernatant. We're going to make a vegan milk from that by combining the casein with a vegan sugar and a vegan fat. And with that milk, we'll use the normal cheese-making process to make a real vegan cheese. So there's 4 major cheese proteins called caseins: alpha S1, alpha S2, beta casein, and kappa casein. These proteins coagulate to form a structure called a micelle. We are also looking into an enzyme called chymosin FAM20C, which phosphorylates casein and is important for micelle formation. These micelles have hydrophobic tails that stick outwards. These hydrophobic tails keep the micelles from sticking together, which keeps milk a solid. However, in cheese, the hydrophobic tails are cut off, and the micelles clump together, forming a gelatinous network. This is a basis for cheese. We will combine these casein micelles with vegan sugar for cheese ripening bacteria and a vegan fat. Here are our milestones. So our first milestone was to design our parts, which we have accomplished. Our second milestone was to clone and transform the plasmids into E. coli and confirm the sequence. As of October 11th, we've confirmed 100% of the constructs. Our third milestone was to transform the plasmids into yeast. We're currently troubleshooting optimizing our yeast expression system. Our fourth milestone was to verify protein expression and secretion in yeast and purify proteins. We're currently going to work on this. Here's our part design. We've codon optimized the target gene, in this case, casein or kinase. We've attached an alpha factor secretion tag, which is cleaved off during secretions by the KEX2 protease sites. Where necessary, we have eliminated internal KEX2 protease sites. We have flanked it with SAP1 sites for cloning into the electrovector, and we've also flanked it with a bioreg prefix and suffix for submission to the registry. We have synthesized 11 count human casein and kinase constructs as IDTG blocks. We use the Electra PD1214 plasmid backbone. Some advantages are that it's IP-free and has 5-minute cloning, scarless cloning. We transformed it into E. coli, and there's something really interesting revealed by our sequence confirmation on October 11th. It was revealed that kappa casein, KEX-, may be toxic in E. coli. However, 2 of these biobreaks, bovine beta casein and bovine kappa casein, were sufficient for micelle formation. We then transformed the plasmids into yeast. We used an oxytrophic yeast strain, Ura-, which is unable to make uracil. The PD1214 plasmid has a missing Ura-3. We're also using 5-FOA, which will kill yeast with intact uracil pathways. This can be used to counter-select against wild-type yeast. We are currently troubleshooting yeast contamination issues. To deal with this problem, we've swabbed numerous surfaces in the lab, including our benches, drawers, equipment, bioreactors, water sources, and more. You can see some of these in the image here. The PD1214 plasmid carries a strong constitutive TEF yeast promoter. Our construct contains a yeast alpha factor that should target the proteins for excretion. In the ideal case, we should see a thick protein band on the SDS page gel of the supername. Additionally, expression of kappa casein can be verified by cleaving with chymosin. We can precipitate the casein proteins by changing the pH and calcium ion concentration. This is our work in progress.
So as Andrei mentioned, this is still very much a work in progress. So can we actually pull this off? It's important to keep in mind this is actually the first attempt at making a bulk food from recombinant purified protein. So I'm sure there will be a bunch of hurdles to overcome scaling this up. We're targeting a yield of around 2.5 grams per liter, and that's based on the existing published work expressing a partial casein. So when people ask us how much is it going to cost, I tell them, well, think of 5 grams of cheese for the price of a liter of very expensive beer. So that's the price target we're aiming for. So compare that with Mark's Post's work on his in vitro hamburger. The first hamburger he created cost $250,000. So we're definitely going to be orders of magnitude below that. So we're hoping to produce tastable amounts of protein within months and planning for having an actual product sometime in 2016. So this is not just a summer project for us, and that's important to keep in mind. This project goes well beyond the iGEM, and we're in this for the long haul. We actually set up a nonprofit corporation to shepherd this project further. We actually started out, as you saw, with a large Indiegogo campaign, raising over $37,000. A big chunk of that is actually earmarked for scale-up experiments. So $10,000 is going to go for a large 50-liter bioreactor where we can actually start making protein in significant amounts. And one of our stretch goals was that we're going to make narwhal cheese, because we can. The narwhal genome is currently being worked on at Santa Cruz. And if we can make narwhal cheese, we're one step closer to making unicorn cheese. So our Indiegogo campaign was actually a really useful tool for us in terms of outreach to our community. We've gotten a lot of feedback through that. We've had more than 51,000 visits to the page, 700 tweets, 8,000 Facebook shares. We have almost 700 paying supporters from 20 different countries. One of the rewards that we were offering was Ask a Biohacker, where people could send us a question, and we promised we would make a video in response. So here you see some of the video responses that we put up. Anything from what's happening, why does dairy cause migraines in me, and is your real vegan cheese going to solve that? To, is it more ethical to like robots or dinosaurs? And of course, our answer is narwhals. We also had a reward level, a $2 level, tell us why we're wrong. So we're inviting our critics from expressing what problems they have with this project, and we were promising a written response in return. So we have actually replied to a bunch of those either to our comments section or through email. So we've had a lot of publicity coming out of this, featured in over 100 articles. We actually did a very extensive Reddit Ask Me Anything, where we had like constant sort of questions and answers throughout the entire day. So you see some of the quotes from some of the articles. I'm sure synthetic biology can get weirder, but this is a great start. I'm looking forward to tasting some. We've actually had tons of feedback from the community, and especially from the vegan community who's really in favor of this. We were kind of worried whether the vegan community was going to align with sort of vegan, organic, non-GMO. It turns out that most of the vegans we've talked to have been overwhelmingly in favor of this project. So reaching out to the community is actually really important to us, and we've built in lots of mechanisms to provide that.
And that stems from our approach at creating community labs. We are public from the start. There's no sense of us and them. Whether you come from a walk of life that is professional or engineering or scientific or completely other, you're welcome in our lab. And we took that approach from the start with our team. First, deciding whether even to do a team in a grassroots way, bridging from the communities already built at Counterculture Labs and BioCurious. We're very lucky to be in the Bay Area, where we have perhaps the largest community of extra-institutional biologists in the world, with a reach of over 2,300 people. And we used relatively free or easy-to-use technical tools, which anyone can access to broaden our outreach, including meetup.com, where we posted every single meeting that we have held. And in addition, we're still posting our meetings on Meetup. We have open membership on Google Groups with 84 members. We even wrote articles for open access publications, invited the media further to participate in our meetings. We convinced them to work in the lab alongside of us. And we went to Maker Faire, where we earned 2 awards, Best in Class and Editor's Choice. And I want to emphasize that we use our working groups as a way to broaden public engagement. So we do a roundtable introduction of each person, where they're from, what they're interested in, and we help encourage them to go into different working groups or allow them to self-select. As community labs, we have to think a lot about why we exist and what we support and protect. And we have namely 4 groups that we're working for. So that's our lab members, our community and visitors that come to the lab, and the broader local environment and the world. So we know that what we're creating and what we're about, the implications extend much further beyond our lab, our biohackers, and our cheese. So we're working on lots of different work in this arena.
So I'm going to talk a little bit about the public perceptions of synthetic biology, because some potential concerns that people might have with our project, because we share many of the same concerns, so we want to take them seriously. As background, these are some results from a focus group that was done on synthetic biology, and it shows that participants were really supportive of applying synthetic biology to treat disease or clean up the environment, but they had a lot of concerns when it comes to using synthetic biology to create chemicals or food products. So what might some of these concerns be? One thing that scholarship in STS shows is that people are not generally opposed to the use of a technology like genetic modification under all circumstances, but instead they often raise concerns about the context in which technologies are developed and tested. This includes concerns about whether institutional actors are really serving the best interests of the public. We think our project has the potential to change the conversation in this area, because we are actually changing the context in which food biotechnologies are developed. So we're a nonprofit that's dedicated to open science in the public benefit. Patrick and Ari have already talked a little bit about what that means. All our meetings are open to the public. It also plays out in how we think about issues like intellectual property. So we've consulted with intellectual property lawyers to talk about how best to ensure that our methods do really remain free and open for anybody to use. Another potential concern that people have raised with respect to GMOs in the past is the potential for harm to the environment. And we think it's significant that this project is environmentally motivated. So dairy the dairy production causes pollution, it causes deforestation in many countries, and it's a major contributor to global warming. Cows produce a lot of methane, and in fact, 2.7% of the total greenhouse gas emissions come from milk production. So we think that the method of producing casein and yeast could potentially be a more environmentally sustainable way of getting to cheese. We've done some rough estimates. A lot of work went into these very rough estimates, but we think that we could potentially reduce greenhouse gas emissions by 40 to 90% per gram of casein. And having a better vegan cheese substitute could also help more people switch to a plant-based diet. But at the same time, we also have to make sure that we don't cause any unintended consequences to the environment. So we're also, there are several ways in which we're taking into account this health and safety of people and the environment in the design of the project. So for one thing, our cheese won't contain any GMO. We plan to purify the protein before we make the cheese. We've also chosen a host organism that's already eaten every day, baker's yeast. And we're using a technique that's actually already used in cheese production. So about 80% of cheese on the market now is produced with chymosin that's derived with fermentation from genetically modified organisms. The traditional method is getting it from rennet taken from the stomachs of cows. And so we're also, another thing about this project is that it doesn't involve any release of genetically modified organisms into the environment. We plan to contain the yeast in a bioreactor, and as guards against unintentional release, we're using an oxotrophic strain and good biosafety techniques. And finally, we're consulting with the FDA in order to make sure we understand how to ensure that any cheese that we produce is really safe for human consumption. And we've also reached out to some public interest groups and NGOs that have expressed concerns about synthetic biology to see if we can start a dialogue around their concerns. So to sum up, our project is changing the conversation around synthetic biology in these ways. Education is part of what we do, but instead of starting from the assumption that the public has a knowledge deficit that needs to be remedied, we acknowledge that everyone has expertise in a different area, and we encourage community members to get involved and contribute what they have to offer and learn more about what they're interested in. So in these kinds of projects, the question that we're asking the public changes from should scientists or corporations be allowed to do X to how should we use these tools together. In sum, we're not seeking to change the public perception of synthetic biology. We're seeking to change the future of synthetic biology through wider participation and engagement. Thanks.
So I have two questions. One of them is actually about the yeast contamination issues that you were finding. And if you could maybe talk a little bit more about that, and how it impacted sort of your project moving forward, and how you possibly see that impacting sort of some of those safety issues that the FDA might be concerned with. And then my second question is kind of a more broader sort of public perception policy question. You know, I hear you guys have said this before. It's like, you know, why are you doing this? And you say because we can. And so I wonder if you can address sort of a larger symbiotic question maybe too. Is this well, if you should?
Yeah. Well, let me maybe take the first question. So dealing with the yeast contamination, it's all part of sort of the education process and community team, because we're getting a bunch of people that have never done any biology before. So in typical student team, hopefully the students might have had like a semester of molecular biology or something, or you can push them through an actual formal course. We're teaching people from scratch as they go along. And sometimes we have to go back and sort of go over sort of basic sterile technique. And so we did this exercise of swapping everything inside and showing where the contamination was and how people might be tracking it. Obviously, eventually, this will be produced in an industrial setting. And once we actually talking about scale-up, we will probably partner with an actual cheesemaker that has sort of the correct food processing facilities. So the second question, do you want to, what was the second question again? Sorry.
So it's this question of, you know, because we can, which is kind of what the answer usually is with some, you know, kind of, I think, cool project like this. But I think a lot of people would ask, you know, well, should you? Not if, just because you can, but whether you should do it.
Yeah, I would say that this project is not motivated just because we can. It's motivated by community members that have long traditions in activist communities and really care about issues like the environment and animal rights. And I would say those are the main motivating factors. But then, yeah, we do spice it up a little with the narwhal cheese.
Yeah, actually, sort of this issue of cheese is one of the major reasons that we've heard from vegetarians as reasons for not switching to a fully plant-based diet. So the more we can help people switching to eating lower in the trophic chain, the better for the whole planet.
Okay, thanks guys. My question is about your genetic designs. So can you talk a little bit more about the thought process? Are you expressing the proteins together or separately? Why did you choose the TEF promoter? Can you tell me a little bit more about that?
So most likely, we will probably have separate strains of yeast, each with producing a single protein, because we might need to optimize the production process for each protein individually. So in an industrial scale production, you would probably make each of the proteins individually and then combine them to make micelles. That's the most straightforward approach. So it means we don't actually have to make very complicated constructs, and we can actually have each construct on a single plasmid. So that's what we're working with right now.
And my follow-up question is, where does the expertise come from in a community lab? Are you all self-taught, or are some of you trained as biologists?
So we have a number of people involved that are trained in biologists and that have done postdocs at Stanford and other places that know sort of everything about how to do the molecular biology side. We have people that are more involved in thinking about sort of the ethical, legal, societal issues. So everybody comes with this project with their own levels of expertise. So people sort of talk about amateurs in terms of DIY bio. Most of the people we get are actually experts. They're not necessarily experts at doing synthetic biology, but they carry their own expertise with them.
Answer that a little further. Some people are almost insane about how much work they're going to put into this, and if they're really motivated, they're going to learn in very similar ways to how someone might learn at university, however in an applied way and on a fast track, because we have some already foundational work done on our project before they step in. So I believe the project was motivated by a vegan scientist as part of our community, and he had been thinking about this for years, so he had some ideas in mind, and we knew in a fairly straightforward way what we needed to do. And this sort of scientist mentor and anyone else who wants to participate, if they're willing to do enough, spend enough time, is this mentor-student model that happens pretty organically at our spaces very frequently.
And we see that pretty much everybody has something to teach and something to learn. So it's really more peer education rather than a classical mentor-student.
We take a quick question from the audience. Yeah, so you mentioned on the Indiegogo campaign that you had people, it's a basically great project, yeah, amazing. Yeah, so on the Indiegogo campaign you mentioned that for $2 people could say why you shouldn't do it. So what kind of things did they say and what kind of responses did you give to those? And can I see them online somewhere, maybe?
Yeah, most of those responses are in the comment section on the Indiegogo campaign. Some people contacted us through email anonymously, so or wanted to stay anonymous, so we just responded to them through email. Some of the questions we got was, doesn't casein cause cancer? So we actually spent some time digging into literature around that, and that seems to be very poorly supported. So that's not something we have concern with. Or some of the other writing. GMOs are evil, the usual. Right? so it's a good learning experience for us to sort of how to frame things in a way that somebody who says GMOs are evil will actually connect with your answer.
Thanks a lot. Great. Fortunately, that's all we have time for. We'll have to talk with the team afterwards, but let's thank them again.